Welcome back. Today we're going to be talking about enzymes. Enzymes are mostly proteins that help us perform reactions in the body. Let's take a look at one. Here we see the enzyme in blue. As I said, they're usually proteins, and this particular enzyme is going to bind to the substrate. The substrate is the thing that the enzyme acts on. And in this particular reaction, we're going to take the substrate and we're going to break it down into two products. So we're taking something big and we're breaking it down into two smaller things. Remember that we have already learned the name for that, and it is hydrolysis. So when you break down something big, like a macromolecule, into something smaller, like a monomer, it's hydrolysis, and the thing that's helping us do that is the enzyme. So the enzyme has something called an active site, and this active site is specific for the substrate, the thing that it's going to act on. The substrate fits into the active site, and the enzyme holds it there until the bonds can be broken, and then it's going to release the products. Now, if you notice, the substrate comes in big, it binds to the active site, and then it gets broken down into two products. So it's not the same as when it came and was bound by the enzyme. But the enzyme, this whole time, is the same. So through the steps of the reaction, the enzyme does not change. And it doesn't matter if we're going this way in a hydrolysis reaction or the other way in a synthesis reaction. Remember, synthesis is when we are combining two things to make something bigger. So for example, the substrates here are separate. They're going to bind to the active site of the enzyme. The enzyme's going to hold them together until the bonds can be made, and then it's going to release the product. The entire time, the enzyme doesn't change. So enzymes are very useful because they are going to be able to be used again and again and again. So why do we even need enzymes? Well, enzymes are important because they speed up reactions by lowering the activation energy. And this basically shows you that when you have an enzyme present, the amount of energy that you need for a reaction to happen is much lower, it's here, than the amount of energy that you need when you don't have an enzyme present. And so what we have found is that in the human body, we have a very limited supply of energy. And if we had to put this much more energy into every reaction that happens in the body, either synthesis or hydrolysis reactions, we wouldn't have enough and the reactions wouldn't happen fast enough. So you might be wondering how an enzyme can actually lower the activation energy. Well, it helps to hold the substrate together. Either puts these two substrates sort of together in the right area so that they can bind correctly, or holds them together and then acts on the bonds that are already between them to break them up. So it kind of helps to stabilize that substrate until the reaction can happen. Sometimes the reaction can happen without an enzyme. It just takes a lot more energy and a lot more time. And we just don't have that kind of time to wait for that to happen. Quick review. Enzymes are mostly made up of protein, sometimes RNA. For example, ribosomes are made up of RNA and proteins. They help to speed up the rate of reaction. Enzymes are not used up or altered in any way in a reaction and can be used again and again. And they are very specific due to shape. For example, an enzyme that catalyzes one reaction between a particular substrate would not be able to bind to some other substrate and do a different reaction. So we have For every reaction that you want to do in the body, you have a specific enzyme for it. And the substrate is going to fit and bind to the enzyme in the enzyme's active site. That's it for today. See you in class.